Let us go over the very first lab that we did, and that was our osmosis, and diffusion. So in osmosis and diffusion, we have learned how do particles move. And we agreed that particles do not move except as a result of some form of energy that is causing them to move. There is heat energy that increases the average velocity of particles, causing them to bombard with each other, to collide with one another, and they will move. They will not move in a random direction. They will move from an area of a higher concentration to an area of a lower concentration. So this is known as diffusion. Diffusion is not powered by ATP. It is not powered by electricity. It is powered by the particles colliding with one another. That is caused by kinetic energy as a result of heat. We have also learned about Brownian movement. And Brownian movement is called just that because it was discovered by this scientist by the name of Brown. And he realized that when he dropped from a dropper in a solution of water, he dropped some dye, he realized that the movement of the dye was not a straight line. Although his hypothesis said that the dye should move in a straight line. But he realized that this line was curved, and it showed that it was zigzagging. And he realized that is because the collision and the bombardment movement of the solvent molecules with this dye. So these molecules are bumping with one another, and that represents the nature of that movement. If we have a membrane, that divides to us two sides, the membrane divides to us two sides, A and B. On side A, I put 500 millimolars of glucose, and on side B I put 100 millimolars of glucose, and I convert my barrier, my membrane, into a semi-permeable or a differentially permeable membrane that allows for the movement of glucose. Glucose is really big. Is bigger, a lot bigger than sodium. Sodium is an electrolyte, is an atom, is an element, Na. But glucose is a molecule composed of many carbons, and these carbons are attached to an oxygen. So this molecule is huge, it cannot go through the pores. But if we have pores that are large enough that allow the passage of glucose, what is naturally going to happen is that the movement of glucose, there will be a net movement of glucose through the semi-permeable or the differentially permeable membrane from an area of a higher concentration to an area of a lower concentration. That's half a point. Acknowledging the law that glucose is going to move from an area of a high concentration to a low concentration. The second half is that movement is not going to go forever. You're not going to have the entire 500 going to the 100 side until you have 600 on one side and 500 on the other. The movement will happen until they equilibrate. They will reach equilibrium, and that will be the sum of both 500 plus 100 divided by 2, 600 divided by 2, that is 300 millimolars of glucose. 300 millimolars of glucose. That is if the membrane is semi-permeable, differentially permeable, allowing for the passage of my glucose. Now that shows us diffusion. This is not osmosis. This is diffusion, which is the movement of particles. Diffusion, which is the movement of particles from an area of a higher concentration, from an area of a higher concentration of area of a higher concentration area of a higher concentration of solute to an area of a lower concentration of solute. That is diffusion. Osmosis is a different case. So water itself has the property of osmosis. So, by the way, when I say glucose is 500 millimolars and on the other side is 100 millimolars, some students think that the 500 millimolars is depicting the size. So the one on side A is so big, like somebody who is 500 pounds and another person who is 100 pounds. No, that does not, millimolar does not represent the size. That represents the quantity, the number. There's like 500 people on one side and 100 people on the other. So this is not describing a molecule of glucose. That's describing their quantity, their number, their amount. And the same thing with water. So if we replace side A and side B with 500 milliliters of water, 500 milliliters of water, and 100 milliliters of water, and you open this membrane to allow the passage of water. Water will start moving again from an area of a higher water concentration, from an area of a higher H2O concentration to an area of a lower H2O concentration until they equilibrate at 300 milliliters on each, 300 milliliters on each side, and then they will equilibrate. What is that an example of? Is that an example of diffusion or osmosis?
Diffusion.
That's diffusion. Why diffusion?
Because osmosis is the diffusion is water.
So that is water. H2O.
Because osmosis happens in the cell, within the cell, no?
Yes.
Doesn't osmosis go from low to high concentration?
No. From low water concentration to a high water concentration. This is diffusion. Osmosis is when water moves from an area of a low solute. So here I have no solute. That's just pure water. But if I add glucose here, I add 100 millimolars of glucose on side A, and I add 50 millimolars of glucose on side B, and I'm not done because I have to also mention that the membrane is impermeable to glucose. It does not allow for the passage of glucose. So now who will move? If the membrane is impermeable, if the membrane is impermeable to glucose, in this solution with water and glucose on both sides, who's going to start moving? This membrane in the middle is not allowing for the passage of glucose. It's only going to allow who to pass? Water. Now water will move from an area of a low solute, low glucose concentration, 50, to an area of a high solute concentration, 100. Why is that? Because water has very important properties known as water has high specific heat capacity. It has high specific heat capacity, and we use that for sweating and thermal regulation. And water is very important because it is a versatile solvent. It's a versatile solvent, and it helps. It helps dissolving. It is the solvent that is more likely than any other solvent to dissolve every solute out there that is polar. So who is the champion of being able to dissolve the most amount of solutes? It's definitely water. And that is what drives osmosis. So in osmosis is a form of diffusion, but it is not a diffusion from an area of a higher concentration gradient to a lower concentration gradient. It's the movement of water from a lower solute. So now the solute cannot move. The glucose cannot move through this semi-permeable or this differentially permeable membrane. It cannot move through it. So on its behalf, water is going to move from an area of a low solute concentration to an area of a high solute concentration.
But that's still not gonna give you like a balanced amount of glucose. So what's gonna happen? The water.
No, the water is going to move until it breaks glucose apart, and that will make the osmolarity equal. So the equilibrium here is not going to be based on concentration. It's going to be based on osmolarity. And something that you really need to understand regarding osmosis, and that's going to definitely be on your exam, is what is the fate of a red blood cell that is put on 3 different solutions. So the first solution, so we have 3 beakers, beaker A, beaker B, and beaker C. And they are all full of a solvent. So the solvent is more than the solute, and the solvent is the one who is dissolving the solute. So in this solvent, you put 3 red blood cells. 1, 2, 3. On one of them, let's say that the red blood cell osmolarity is 300 milliosmoles. So it's standard. You cannot change the cell osmolarity. So the osmolarity of your red blood cell is 300 milliosmoles. 300 milliosmoles. So on beaker A, we're going to have the solvent 1000 milliosmoles. So here the solvent is more. The osmolarity of the solvent is higher than the osmolarity of the cell. So the osmolarity of the solvent is much more than the osmolarity of the solute. And my solute here is going to be my red blood cell. So what kind of solution is that? What kind of solution is that? This is a hyperosmolar solution, also known as hypertonic. Also known as hypertonic. For B, we can have 300 milliosmol solution that is equal to the red blood cell. So here the osmolarity of the red blood cell is equal to the osmolarity of the solvent.
Is it an isotonic?
That's isotonic, like an isosceles triangle. So that is an isotonic. That's an isotonic solution. And lastly, you can have the red blood cell with 300 milliosmoles and the solution is only 100 milliosmoles.
Hypo.
So here the osmolarity of the solution is less than the osmolarity of the cell.
Is it in hypo?
So that is an example of a hypoosmolar solution or hypotonic. That's a hypotonic. So for the exam, you have to be able to tell what is a hypotonic, what is a hypertonic, what is an isotonic solution. Then you also have to tell what is the fate of the red blood cell. So we said that according to osmosis law, the water will move from an area of a higher water concentration to an area of a higher solute concentration. We're going to move from an area of a low solute concentration to an area of a high solute concentration. So here the water is going to get out from the red blood cell into the solvent, and that will cause the red blood cell to shrink. Red blood cell is going to shrink. Then in the isotonic solution, there will be a steady state. There will be a steady state. There will be a steady state of water movement to the inside of the cell and outside of the cell. So the rate of water entering the cell will equal the rate of the water exiting the cell, and that is a equilibrium. In a hypotonic solution, the solvent is less osmolar, is hypoosmolar compared to the cell. So what happens here, water is going to start flowing to the inside of the red blood cell. It's going to start flowing to the inside of the red blood cell, and that will cause the red blood cell to swell. So you have to know, does it swell in a hypotonic or a hypertonic or isotonic? Does it shrink in a hypotonic, hypertonic, or isotonic? I'm recording that. I'm going to post it, so you don't have to waste your phone memory. I mean, if you want to, you can do that. But I'm going to post it for you anyways. Okay, so that's very important for diffusion and for osmosis. Another thing is, let's say that I have a beaker. Let's say that I have a beaker, and this beaker has, for example, 9 milliliters of water. The beaker has 9 milliliters of water. That is my solvent. And 1 milliliter of sucrose. And that is who's the smallest guy? The big guy is the solvent. The small guy is the solute. So 1 milliliter of the solute. Now on the exam there's a question. If I add 1 milliliter of water, what's going to happen to the sucrose? What's going to happen to the solute? It's going to decrease its concentration. So before we had 1 milliliter of sucrose dissolved in a 10 milliliter solution that is 9 milliliters of water and 1 milliliter of sucrose. If you add an extra milliliter of water, so now you have 10 milliliters of water. So what happened to the sucrose? It went down. So if they're telling you that we added water, you immediately conclude that the solute went down. And if they tell you on the exam, the question says, we add more solute, that means the water went down. They are not in a seesaw. They are dissolved in a locked solution. They are like in a seesaw. So you know the seesaw in the park, like this. In the seesaw in the park, this is the solvent. It's the heaviest. That's why it's down. And it's a solute. It's up in a beaker. If you add more solvent, then the solvent will increase and the solute will decrease. Its concentration will go down. But if you increase the solute, that means the solvent is going to decrease. Because remember, 9 milliliters of water, 1 milliliter with the pipette, 1 milliliter of sucrose. If you add another milliliter of sucrose, water went down. Because it's one beaker. You're not opening a faucet, getting more water. You're not getting more distilled water. So the more you add of either, they are like playing a tug of war. If you add one on each side, the other one is going to get weaker. It's going to get less. So you add more solvent, the solute is going to go down. You add more solute, the solvent is going to go down. Because it's locked. It's locked in one state. You add more solvent, the solute is going to go down. The solvent is going to go up. If you add more solvent, you have solvent and a solute in the same solution. Solvent and solute in the same solution with a ratio. The ratio is 9:1. If you add 1 milliliter to the solvent, the solvent is going to go up, but the solute is going to go down. Solvent goes up and the solute goes down. But if you add more solute, the solute concentration is going to go up and the water concentration is going to go. Okay, you're not paying attention. Okay, the RF. RF is very important. That is going to be part of our chromatography experiment. Then after that, we went into the enzyme. What you need to know about the enzyme, the key point that I'm going to ask you about on the exam, is that what are the properties of the enzyme? The enzyme needs optimal conditions in order for it to operate. It needs an optimal pH and it needs an optimal temperature and it needs an optimal enzyme concentration and an optimal substrate concentration. So let's see the properties. Substrate concentration, and enzyme concentration. Okay, so let's see what happens. Enzymes are proteins. They are made up from amino acids. They are not made of sugar. They are not made of lipids. They are not made of nucleic acids. They are made due to the translation of the amino acids, the RNA, but they are amino acids that are folded upon one another thanks to the hydrogen bonding and hydrophobic effects and ionic bonding, and then we fold up to go from primary to secondary to tertiary to quaternary. Then these enzymes, if you do not give them the proper temperature and the proper pH, they're going to denature. They're going to denature. Denature meaning they disintegrate and they crumble and they fall apart. Because they bind to one another, as I just said, due to the folding. Folding. What are the bonds that make the folding happen within your amino acid sequence? These are your hydrogen bonds. These are weak bonds, not covalent bonds. Hydrogen bonds, hydrophobic effects, and ionic bonds. So if you change the temperature, if you mess the temperature up, these weak bonds are going to fall apart, leading to my protein enzyme to disintegrate. So most of the enzymes are proteins. That's very important for the exam, that you know what the nature of these enzymes. Are they lipids? Are they sugars? Are they nucleic acids? No, no, and no. They are amino acids, and that are folded to give us macromolecules known as proteins. So proteins, they have the proper temperature. If you change the temperature, they're going to, if you heat it up too much, they're going to denature, because now you're going to destroy the hydrogen bond, the hydrogen bonds, and you're going to destroy the hydrophobic effect, and you're going to destroy the ionic bond. And then the enzyme, who is usually a quaternary structure, is going to fall back into a tertiary structure, falling back into a secondary structure, falling back into a primary structure, and falling back, the arrow points backward, meaning that this is a denaturation. It's going to denature. That is denaturation. Denaturation. Okay. Now, what about the pH? Enzymes have different pHs, because some enzymes work in the stomach where the pH is really low. Like hydrochloric acid has a pH of 1, that's very acidic. So these enzymes that are working in your stomach to digest the food that we eat, they working under very low pH. Whereas another type of enzyme, for example, like the enzyme that digests sugar in the oral cavity in your mouth, that needs a different pH, 7. So these are two enzymes doing something similar, catalyzing. Enzymes are biocatalysts. Enzymes are biocatalyst. What does biocatalyst mean? Biocatalyst means that it increases the rate of biological reactions. So not all enzymes catalyze biological reactions. Some catalyze chemical reactions, but the ones that we are concerned with in biology, these are biocatalysts, and they need their optimal pH and optimal temperature. When it comes to enzyme concentration, the relationship is linear. So the more enzyme you add, the more the reaction is going to go up in a straight line. So if this is the reaction rate on the x-axis, that is my reaction rate on the x-axis, and on the y-axis is my enzyme concentration. The more enzyme I add, the faster the speed of my reaction. I have a lot of people waiting outside looking for a shuttle bus. The more buses come, the more people gonna ride, the more trips we're gonna have. So the rate of people being shuttled is gonna increase with the more number of shuttle buses. But the problem is, that's not the same if you look at it the other way, where you have a fixed number of shuttle buses, like enzymes. You do not increase the number of enzymes. You do not increase the number of shuttle buses. You increase the number of people who are trying to get on a shuttle bus. So in the beginning, shuttle buses are empty. We have like 5 or 6 of them, and you have 700 people coming. So at the beginning, 50 people get on the first bus, 50 on the second bus, 50 on the third bus, and then the buses are gone. The 3 buses are gone. So in the beginning, so the relationship is not linear. It's only linear with the enzyme. It's only linear with the shuttle bus. The more enzymes you add, the more shuttle bus you add, the more trips and reaction rate is gonna go up. But when you increase the substrate, in the beginning it's gonna go up until all the enzymes are fully saturated. Then you're gonna level off. You're going to level off, and you're going to have a ceiling effect, because you have reached saturation. saturation. All the enzyme active sites have been saturated, so the reaction rate is gonna stop. No matter how much more substrate you add, you're not gonna have a reaction. No matter how many people coming out the subway, nobody's gonna go nowhere because all the shuttle buses are full. So it's very important for you to understand what are the effects of increasing substrate concentration and what are the effects of increasing enzyme concentration. Then you have to look at the graph and you have to know the difference between activation energy and free energy. Students do not know the difference between activation and free energy. Okay, so let's look at this graph for example. This graph shows us that as a function of time, time is always on the x-axis, that is time, that is always my independent variable. It does not change, it is independent, and this is my dependent variable, and that is known as delta G, Gibbs free energy. So in order for me to go from reactants, if I want to go from reactants to products, I have to go up a mountain and then come down. This mountain, on the top of it, I have a point that's very high known as the saddle. That's my saddle point. That's the saddle point. This saddle point is very important because that is the point where you're going to have your transition state. Your transition state complex. This transition state complex is an intermediary. It's an intermediate that's going to be right at the top of the saddle, like a horse saddle. It's going to be right up here. Right up here. That's the transition state complex, TSC. So the activation energy is the energy from the reactant up until the transition state complex. You see it's really high. But the energy between the reactant and the product here, that is the free energy. That is the free energy. That is the net free energy. So the net free energy will never change because the enzyme is going to bring down the activation energy. It's going to say, you don't have to take the straight line, A. You can take the dotted line, B. And I'll give you an alternative activation energy. So you don't have to go up through a mountain. You just have to go up through this little hill. So what does the enzyme do? It brings down the activation energy. It does not affect the free energy. So usually on the exam, the tricky question is when you tell a student, the enzyme is a biocatalyst who is able to accelerate the rate of the reaction by bringing down the free energy barrier. True or false? True or false? False. You see, it's false because it only brought this guy down. This one is always unaffected. That's like if you have to pay a certain amount of money. That is what it's gonna worth. You have to pay that much. This guy is going to get you to the Apple store faster. It's going to do things to help you, but it's not going to pay your bill. You gotta pay the bill from going to reaction to product. That is your net free energy. The net free energy is always the same and nobody can change it. We only can change the activation energy, which is the energy between the reactant and the... No. The reaction and product, the energy between them is the net free energy. I'm talking about the activation energy. What is the activation energy?
The reactant and the saddle point.
Excellent. And who's on the saddle point?
The TSC, no?
Yeah, what's the TSC? Very good. Transition state complex. So what's the transition state complex? Transition state complex is, let's say my reaction says AB plus C. I must have an exchange and end up with AC plus B. On the transition state, you're going to have an intermediate, where A is going to be bonded with B and bonded with C. The three of them are bonded weakly. So you're not a reactant, but you're not a product just yet. So you are in the intermediate, in the middle. The energy going from reactant to the saddle, that's the activation energy. So the enzyme gives us an alternative transition state complex. That's how it's able to bring down the energy reaction. Say you don't have to you don't got to take the subway all the way up there. I'll take you with Uber through the FDR, and that's what it does. But it's not going to change the distance. It's going to change the path. You don't got to, if you want to go to Queens, you don't have to go all the way from Brooklyn through Manhattan, then into Queens. It's going to say, I'll take you from the back using the tunnel. But the displacement is the same. That's the net free energy. You still have to travel that same distance. If you take a helicopter or if you take a subway, the distance is different, but the displacement, you can never change it. From LIU to NYU and there, that the displacement is the same. You cannot make that shorter. But the route you take is you're gonna get there faster. Is that clear? So what did the dialysis membrane represent? When we did our dialysis experiment, what did the dialysis membrane represent? That represented the cell, because the dialysis membrane, how did the dialysis membrane represent the cell? It was similar to what?
Because it had a semi-permeable membrane?
Yeah, the plasma membrane. So it was similar to the plasma membrane. So what happened was when we added the dye was able to move from the solvent into the inside of the cell down concentration gradient. So that showed us we move with concentration, with collision. People are going the same way people bump into each other and move to an area that is more spacious from an area that is really crowded with a high concentration to an area that's more spread out. Then when we did the enzymes lab, we went over Guaiacol being able to detect. Guaiacol is able to detect for us the presence of the product, and that was a marker for the activity of peroxidase. So the reaction was hydrogen peroxide, okay, plus the catalase, who in this case was, who was our catalase? Peroxidase. Excellent. That was our catalase. And peroxidase is going to convert H2O2 into water and oxygen. We're going to use water for blood volume, for osmolarity, osmotic balance in our body. Water is very useful. It nourishes our tissue. And why is oxygen important? Oxygen is important for oxidative phosphorylation and aerobic respiration. So then we add Guaiacol. What Guaiacol is going to do is going to detect oxygen, and Guaiacol is free and colorless. But when it finds oxygen, it's going to convert into amber brown color. So we used that as a marker for the activity of the catalase enzyme peroxidase. So we change the temperature, we change the pH, we change the concentration of hydrogen peroxide, which is my substrate, and we change the concentration of my enzyme peroxidase, and that is known as my enzyme. The enzyme has an active site in which the substrate binds to, and it also has an allosteric site. The competitive inhibitor binds where? It binds to the active site. it competes with the substrate. But the non-competitive inhibitor, it binds to the allosteric side from the back door. And once it binds, it causes conformational change, preventing the binding of our substrate to the active site. So that one is important to know. So we changed hydrogen peroxide concentration, and we realized you're going to get this, because the more substrate you add, the reaction is going to speed up. Like when you add more people to the shuttle bus, the bus is going to start moving, and then when you have like 1 million people, then it's going to level off. It doesn't matter how many people you have, because your buses are all full. But with the enzyme, it was not like that. It was a straight line. The more peroxidase you add, the faster the reaction is going to go. The temperature. If you increase the temperature beyond the optimal, you're going to denature your enzyme catalase. The pH. If you mess up with the pH, we know that the amino acid is made up of an alpha carbon. This alpha carbon has 4 terminals. It has a hydrogen terminal. It has a alpha carbonyl with an OH. It has a nitrogen amino terminal, and it has the R group. The carbonyl terminal, that is acidic, and it's gonna donate hydrogen proton if you need it in your body. That's why amino acids are a buffer system. While the amino terminal, these are basic, and they're going to accept hydrogen protons when you're suffering from acidosis and there's excessive amount of hydrogen protons in your body. So I want us to now go over the photosynthesis lab. And remember, for photosynthesis, you have to understand that we have two different and two separate reactions in one. The same way that we had 2 different reactions in one. That's why that photosynthesis formula is not telling you exactly what happened. It's summarizing everything. That peroxidase reaction just showed us in one reaction what is actually happening is the 2 reactions. The first one was the catalysis of the oxidation and the breakdown of hydrogen peroxide, muting it from a reactive oxidative species byproduct into water and oxygen. And the second reaction, when Guaiacol came in and detected that oxygen and changed color from colorless into amber brown. In photosynthesis, you have 2 reactions. The first one is the light reaction, and the second one is known as the dark reaction, or the dark phase, known as the Melvin-Calvin reaction or cycle. And what happens is, in the light reaction, we take sunlight energy, photons, sunlight energy in the form of photons, and these photons excite for us electrons. You know, when people hear music, they start jumping up and down. When little kids start hearing the music, they start jumping up and down. Electrons don't hear the music, but when they feel sunlight, they get excited. So they start jumping up, but they don't come down, they jump up. When they jump up, they collect energy. That is what your chloroplast does. Your chloroplast steal the energy that the electrons got from the sunlight. So there is a reaction center in your chloroplast that facilitates this connection between the sunlight and the electron. It makes sunlight accessible to the electron. And when sunlight hit the electron, like little kids, they're going to start jumping up. When they jump up, that energy is potential energy. They now up the mountain, like the water coming down the waterfall. So you have now photosystems. These photosystems are in your chlorophyll. So these photosystems are going to take electron who jump, now they are at the top of the mountain. They are at the saddle. Remember the saddle? They are at the saddle. And when they fall down, they couple that energy. You know, in a waterfall, when the water is falling down, they put a rotor blade. They put rotor blade, and the water comes down, hitting the blade, spinning the rotor. When the rotor is spin, they put inside a magnet and an electric wire, and then an electromagnetic field is created, and they harvest that energy. The same thing is happening in your chloroplast, by the way. But here we're not using waterfall, we're using electron fall. So we take the energy of the electron, we make it jump up because of who? The sunlight. And when it's up because of the sunlight, it comes falling down. When it falls down, we don't spin a rotor. We bring in hydrogen protons. These hydrogen protons are outside in the stroma. The stroma is outside of the thylakoid. This is happening at the membrane of the thylakoid. Then the electrons, when they fall down, they bring with them hydrogen protons. These hydrogen protons are now shuttled, and there's an influx. They move inside the core of the thylakoid. When they move inside the core of the thylakoid, they bring in with them hydrogen proton. So now you generate a proton motive force. Proton motive force. This proton motive force, there is an apparatus known as the ATP synthase. The ATP synthase. From the name you can tell, synthase. It synthesizes. It makes ATP using that electron motive force, that proton motive force. And the proton motive force is going to spin for me a rotor. That rotor is going to spin and create for me ATP. So now I got ATP from the light reaction. Not just that. That was in photosystem 1. In photosystem 2, I do the same thing. All photosystems do the same thing. They jump up the electron and use their energy when they come down. But photosystem 2 was the first one. It's a little confusing because it's photosystem 2 comes before photosystem 1. So photosystem 2 harvest the energy of, but they do the same thing. They harvest the energy of electrons that jumped up because of the sunlight and came down. Photosystem 2 does that in order to create a proton motive force, while photosystem 1 uses the same energy not for a proton motive force, but in order to create NADPH. NADPH, which is a reduced product from the formally oxidized NADP+. So this is oxidized, and now it's going to get reduced. So now my main products from the light phase are ATP from the proton motive force, and NADPH from the photosystem 2, making for me NADPH. Then the dark phase comes in when we're gonna do, this is the main thing for you to remember, is that the dark phase is your Kelvin cycle. And that is where we do carbon fixation. So when they ask you on the exam, what is carbon fixation? Where does carbon fixation happen? Does it happen in the light reaction or does it happen in the dark reaction? You know it happens in the dark reaction, and that is known as the Melvin-Calvin reaction of the photosynthesis. And here, what is carbon fixation? Is when we make glucose. When we make glucose from carbon dioxide. That's called carbon fixation. Carbon dioxide fixation. We fix carbon dioxide to get glucose. And we don't leave glucose as a monosaccharide. We make a polysaccharide known as starch. So that's interesting how we went from carbon dioxide into glucose into starch. Okay, so please go over, we have went over all these points in lab repeatedly, okay? And you can find them in your lab manual. If you need another trip to the lecture recordings, please go listen to those. And if you want to research and your study guide, I solved it. I didn't ask you the question. I did not ask you any question. Very few. I put like, I think, 3 questions in dark bold, in black bold color, because those ones you really need to know those. But the answer is there. Okay? Make sure that you know all the bullets, every bullet of every lab of our 3 labs. Your entire exam is going to be based on them. Thank you very much, and I'll see you guys on Monday for the exam. Bye-bye.